Hi everyone, welcome back to Useful Genetics. This is Lecture 2H. We're going to talk about the distinction between somatic and germline mutations. This is very important both for thinking about evolution and for thinking about the personal histories of our own cells. So our bodies basically consists of two kinds of cells. Most of our cells are what we call somatic cells. They become the tissues of our body. But some cells are of the germline. They're going to form the gametes. And mutations accumulate in both kinds of cells, but only mutations in the germline can be inherited because they're the only ones that will become part of our gametes. So this photograph is a photograph of a developing embryo. And the embryo formed initially as a zygote, the fusion of egg and sperm, and then that cell underwent many cell divisions and the cells began to specialize. And one particularly important specialization in terms of thinking about the consequences of mutations is the distinction between somatic cells and germline cells. So early in this embryo development, already most of the cells of the embryo, including this little patch that's highlighted here, are somatic tissue. They're going to give rise to almost all the body parts of the adult organism. Only the small group of cells here that are already specialized, already dedicated to one particular fate, these cells are going to give rise to the sexual cells, in particular to the eggs or sperm that this adult is going to produce. Now, as the embryo develops, most of the fetus then comes from somatic cells, and in particular, this patch of cells on the developing forearm limb bud came from this patch of cells in the embryo. Only this very small patch of cells in the developing testes or ovaries came from the germline cells in the embryo. Then in the mature adult, a particularly handsome specimen of adult here, all of the somatic tissues, the head, the arms, the torso, the legs, came from the somatic cells. This patch highlighted in red, this isn't a sunburn, this is highlighting to indicate that this patch of cells actually derives from the same somatic cells in the embryo. Discretely tinted green down here, we have the only cells that come from the germline cells and are going to give rise to the sperm that this man will produce. Now, the roles of somatic and germline cells usually aren't reversible. Once a cell is committed to being germline cells in an animal cell, it's not going to go back and become somatic cells. And somatic cells in an animal normally cannot become germline cells. Now, the new study of stem cells, of pluripotent stem cells, may provide ways to do this, but this certainly is not something that normally happens in normal development. The situation is quite different in plants. Plants don't really have the distinction between somatic cells and germline cells because most plant cells can have descendants that will become gametes. For example, if we were to cut the flower off, or in many plants just in normal development, new flower tissue will arise from what was stem and leaf tissue, and that flower tissue will produce the flower's gametes, the pollen and the ovules, that will give rise to seeds to the next generation. We also take advantage of this if we take a cutting from the plant. For instance, we take a leaf or part of the root and we propagate it, and it can grow into a complete new plant. So the somatic germline distinction really doesn't apply to plants, but it certainly applies to us. Now let's think about the consequences of this for thinking about mutations. If we think about the mutation rate, we've got 6 times 10 to the 9th base pairs. The mutation rate is, depending on how you calculate it, maybe 10 to the minus 9th, 10 to the minus 10th errors per base pair per replication. This means that all of our cells have new mutations. All our somatic cells have mutations. All our germline cells have mutations. In the next lecture, we're going to think about mutations in somatic cells. But for now, I just want to think briefly about mutations in germline cells. So here's a female drawing of a female germline. So we have, again, the zygote. And the zygote divides, divides again, divides again, divides and divides. And eventually, some of these cells give rise to the ovary and give rise to the egg cells. This is the germline. In terms of understanding how many mutations accumulate in a generation from one gamete to the gamete in the next generation, what matters is both the mutation rate per cell division and the number of cell divisions. So in a female, there's about 30, in a female person, there's about 30 germline cell divisions between the ovum that gave rise to the zygote that gave rise to you, and the ovum that you produce that will give rise to your children. The situation is different in men. You remember when we talked about mutation rates, we said that the mutation rate was higher. Most of the mutations that our children inherit arose in their father. Most of the new mutations that our children inherit arose in their fathers. Now we're going to explain why. Here's the male germline. Starts out basically the same. You have egg and a sperm, a zygote, stages of development. Eventually you have the testes, and it's roughly the same number of cell divisions this far. But then there are massive numbers of cell divisions involved in the production of sperm. And this means that on average, from the sperm that gave rise to the zygote here, to the sperm that gives rise to the next generation, there's about 400 germline cell divisions. Now, each of these cell divisions is an opportunity for DNA polymerase to make mistakes. It's like the game that Americans call telephone or telegraph, and the English call Chinese whispers, where a message is passed on from one person to the next verbally, and accumulates mistakes from being not carefully understood. The same thing happens with our DNA. Fidelity is much higher, but there's so much of it to be copied because there's many more opportunities for making mistakes in the male germline than in the female germline. Most new mutations that our children inherit will have arisen in their father and be inherited in the sperm and not in the egg. Now, what about the consequences of these mutations? Well, most of the new mutations that we inherit don't cause us any problems at all. So when the egg and the sperm fuse, they create the zygote. Remember, the zygote is diploid. This will all become much clearer in modules 3 and 4 when we talk about dominance, talk about how different alleles interact. But usually the zygote, even if it inherits a mutation that inactivates a gene, the zygote is usually normal because it's heterozygous. It's got one copy of the normal gene and one copy of the mutant gene, and therefore its phenotype is usually normal. Rarely, if the mutation is directly harmful, then the zygote won't develop at all. Now, that's all I'm going to say about mutations in the germline for now. Much of the rest of the course will be involved with thinking about the consequences for phenotype of differences in the germline. What we've done now, we've raised the issue of there being two categories of cells in our body, somatic cells and germline cells. And only germline cells make the gametes. And these germline cells develop from dedicated germline cells that specialize early in the embryo. Male germlines accumulate a lot more mutations than female germlines because they include a lot more cell divisions. And that's because males produce so many more sperm than females do, something like 300 million sperm a day. That's a lot of opportunities for copying errors. Only mutations in germline cells can affect our offspring. Our somatic cells accumulate lots of mutations, but they're not passed on. Now, coming up next, we're going to talk a bit more about somatic mutations. I hope to see you there. Hi, everyone. Welcome back to Useful Genetics. This is Lecture 2I, where we're talking about somatic mutations. In the last lecture, we made the distinction between germline mutations in the cells that become our gametes and are passed on to the next generation, and somatic mutations in all the other cells of our body, which are not passed on, but die when we die. And here we'll be thinking about how mutations in these somatic cells affect us. We'll think about the mutation rates. We'll think about how the interests of the cells and the interests of the organism can be distinguished, and how mutations that benefit the cell may harm the organism, and mutations that harm the cell may not harm the organism at all. Now, here's our diagram, same sort of diagram we had in the last lecture, of the cells dividing as a zygote develops into a large clump of cells in the embryo. What's different this time is that I've colored the cells different shades of blue to indicate that as the DNA is replicated in each cell division, mutations arise in all the descendant cells, so that even though these cells are all descended very recently from a single progenitor cell, they're already genetically different. Now, what I want us to think about is to think about the fate of a particular cell, ignoring the genetic diversity of all the other cells around it. But before we do that, let's think about the different kinds of consequences that a mutation can have for a particular cell. So in any specific tissue, including in the embryo, some mutations are going to be harmful to the cell. They're going to reduce its ability to grow or its ability to stay alive. Other mutations, probably the bulk of mutations, as we described in earlier lectures in this module, most mutations are in parts of the genome that don't make any functional contribution to the cell, so that most mutations are going to be neutral. They're not going to alter the cell's growth or its viability. It'll be just like all the other cells phenotypically. Finally, some mutations may be beneficial to the cell. They'll increase the cell's ability to grow, to exploit the resources that are around it, or allow it to stay alive under conditions when cells around it are dying. So first we're going to consider the effect of these mutations on the cell and its descendants, and then we're going to consider how these effects translate into effects on the organism. So back to our drawing of a cell in a mass of cells in an embryo. First, if a mutation is harmful to the cell, then the cell is going to be lost from the population of developing cells. It may die, it may just fail to grow when other cells are growing around it. If the mutation is neutral to the cell, then the cell is just going to behave like all the other cells. If the other cells are growing, as they would in an embryo, the cell's going to grow and divide too. If the mutation benefits the cell, increases the cell's ability to grow, then the cell's going to outgrow the cells around it, possibly even to their harm. Now, let's translate that into effects on the organism. If the mutation is harmful to the cell, it probably won't have any effects on the organism. And that's because the death of a single cell in a multicellular organism is usually an extremely trivial event. The neighboring cells will step in, they'll take the place of that cell, take over its responsibilities. We see this even very, very early in development when it's possible to remove single cells. Clinically, it's possible to remove single cells from a very early stage embryo for genetic diagnosis, and the rest of the cells in the embryo take over the role of the missing cell as well, so that the embryo develops into a perfectly normal baby. Of course, the same is true for mutations that are phenotypically neutral. There's usually no consequences for the organism. But if the mutation benefits the cell, enables it maybe to grow when it shouldn't, and to grow into a clump of cells, all of whom have inherited its rapid growth properties, we often view these cells as being precancerous cells. And of course, if the mutation is very beneficial to the cell, allows it to undergo rampant growth, then from an organismal perspective, we describe this as a cancer. Now, the rates of these different kinds of mutations are very hard to predict because different kinds of mutations occur at different rates, and different tissues have different mutation rates as well. So, point mutations occur especially frequently in cells that are dividing. Duplications and deletions, the chromosomal mutations, are generally relatively uncommon, but they have very important consequences when they happen. Mobile elements can be quite common and responsible for a substantial fraction of the mutations in a population. In different tissues, both chromosomal changes and point mutations occur a lot in early embryogenesis because cells are dividing rapidly, and the cell division often makes the kind of mistakes that we'll talk about in Module 10. Tissues whose cells no longer divide often have accumulated large numbers of mutations due to DNA damage that was poorly repaired. And tissues whose cells divide many times, for instance, our skin cells, epithelial cells, the cells that generate, the stem cells that generate the cells in our bloodstream and our immune system, these all often accumulate very high numbers of point mutations because they've divided so many times, just the same as we said in the last lecture about sperm cells. Now I'm going to end with a couple of examples of somatic mutations that you might have seen around you but not recognize them for what they are. So on the left, we have some branches of a magnolia tree in bloom. And you can see most of the flowers in the background, they're all purple. But in the foreground is a branch of cells, all of whose flowers are white. And these will all be descendants of a primordial cell lower down in the growth of the tree where there was a mutation preventing the synthesis of the purple pigment. Another example is seen here in this leafy plant. This is all one plant, and the standard phenotype is to have leaves with green centers and yellow boundaries. But here's a branch of the plant where the leaves are all completely yellow, no green at all. And here's another branch of the same plant where the leaves are all green. And now here's my favorite. This is a tulip, a red tulip, but one sector of one petal, so this is the whole petal, one sector of the petal has yellow pigment instead of red pigment. And that will be because an ancestral cell early in the development of the flower underwent a mutation that blocked the pathway that converts the yellow pigment to red. We'll talk more about pathways producing pigment in Module 3. And that's why this whole sector of the petal is yellow. So we've talked about how Our perspective on mutations is very different when we're thinking about somatic cells. That mutations that can be quite harmful to the cell don't harm the organism, and mutations that actually benefit the cell can be very harmful for the organism because they allow the cell to behave as a cancer cell. Now, coming up next, we're going to talk about mutagens, about events and chemicals that increase the probability that mutations will happen. We'll think of these both in the context of somatic cells and in the context of germline cells. And then in the next lecture, we're going to talk about what of these mutagens should we be worrying about and what don't we need to worry about. I hope to see you there.